Capillary exchange is the topic of the screencast, which you may find in Chapter 11 of your textbook. This screencast was designed to achieve the following objectives. Define capillary exchange. Describe simple diffusion, facilitated diffusion, and vesicular transport of substances across the capillary wall. Describe bulk flow in detail. Capillaries form networks among body tissues to ensure the substances don't have very far to travel in order to enter or leave a cell. The movement of fluid and other substances between the blood of the capillaries and the interstitial fluid surrounding cells is called capillary exchange. If you recall, the wall of a capillary consists of a single layer of squamous epithelial cells and its supporting basement membrane, which forms the tunica intima. In order for fluid or other substances to enter or exit a capillary, they must cross the tunica intima. This figure from your book illustrates the capillary wall. The yellow is the lumen, the interior of the capillary, and the interstitial fluid outside of the capillary is shown in blue. Illustrated here at position 1 is the movement across the wall of the capillary by simple diffusion, facilitated diffusion, and active transport directly through the endothelial cells. Now, thinking about simple diffusion, what type of substances can diffuse across the plasma membranes of the endothelial cells to either enter or exit the capillary? If you said nonpolar or hydrophobic substances, you are correct. So some lipids and respiratory gases such as carbon dioxide and oxygen can move by simple diffusion either into a capillary or out of a capillary because they can diffuse right across the plasma membranes of the endothelial cells as shown here at position 1. Facilitated diffusion, where a transport protein is needed to move substances down their concentration gradient, can occur as well. Charged particles such as ions, sugars, and amino acids can be moved by facilitated diffusion into or out of the capillary. Other substances can be moved into or out of the capillary by active transport. Vesicular transport, shown at position 2, can be used to move large molecules or large amounts of molecules across the capillary wall. Endocytosis transports substances into the endothelial cell on one side of the wall. Vesicles then move across to the other side of the endothelial cell, where the substance is secreted by exocytosis on the opposing side of the wall. Although vesicular transport shown here is transporting substances from the interstitial fluid into the lumen, substances can just as easily be transported from the lumen into the interstitial fluid. Some capillaries have gaps between their endothelial cells, which allow some substances such as glucose or ions to diffuse down their concentration gradient either into the lumen of the capillary or out of the lumen of the capillary. Some capillaries also have large spaces or pores between endothelial cells, which allow movement of large amounts of water and dissolved substances across the capillary wall by a process called bulk flow. Bulk flow plays an important role in the flow of water between the capillaries and the interstitial fluid. Bulk flow results from two opposing forces: hydrostatic pressure and osmotic pressure. Hydrostatic pressure is pressure caused by the physical presence of water. For example, The force that causes water to spill out of a garden hose is hydrostatic pressure. Hydrostatic pressure is the pressure that blood exerts against the inner walls of a blood vessel. Therefore, hydrostatic pressure will tend to move fluid out of the blood of the capillary and into the interstitial fluid. Now that we have discussed hydrostatic pressure, let's discuss the second force involved in bulk flow, osmotic pressure. If you recall, osmosis is the movement of water between two solutions with different solute concentrations. The two solutions are separated by a semipermeable membrane that allows water to cross, but not the solute. Since the solute cannot diffuse down its concentration gradient, water moves to the solution with the higher solute concentration. This is osmosis. The force created as water moves by osmosis is osmotic pressure. Now let's relate this figure to the capillary. Since the protein content of blood is much greater than the protein content of interstitial fluid, water is drawn from the interstitial fluid into the blood by osmosis. And the force that drives fluid into the capillary is osmotic pressure. Now that you have a firm grasp of hydrostatic pressure and osmotic pressure, let's look at their effects on bulk flow across the walls of capillaries. So here we have a capillary. Blood enters the capillary from the arteriole, and blood exits the capillary and enters venules. The purple line here shows hydrostatic pressure. Remember that hydrostatic pressure forces fluid out of the capillary. Hydrostatic pressure is greatest on the arterial end of the capillary, steadily decreases to a minimum value on the venule end of a capillary. Osmotic pressure, shown here on the sort of burnt orange line, is constant across the length of a capillary. Remember that osmotic pressure will draw fluid from the interstitial fluid into the capillary. And again, it's constant across the length of a capillary. Therefore, on the arterial end, where hydrostatic pressure is greater than osmotic pressure, there will be net movement of fluid out of the capillary. This is called filtration. In contrast, on the venule end of the capillary, osmotic pressure exceeds hydrostatic pressure, resulting in the movement of fluid into the capillary from the interstitial fluid. This is called absorption. So again, there is net flow of fluid out of the capillary, or filtration, on the arterial end of the capillary because hydrostatic pressure is greater than osmotic pressure. On the venule end, there is net flow of fluid back into the capillary, or absorption. And this is because osmotic pressure exceeds hydrostatic pressure. Slightly more fluid is filtered from the capillary than is absorbed back into the capillary. The excess fluid is picked up by lymph vessels and eventually returned to the cardiovascular system. Now let's review the objectives of the screencast. Define capillary exchange. Describe simple diffusion, facilitated diffusion, and vesicular transport of substances across the capillary wall. And describe bulk flow in detail. It appears that we have come to the bloody end of this module.